Welcome to the NIHR Dementia Researcher podcast, brought to you by dementiaresearcher.nihr.ac.uk in association with Alzheimer's Research UK and Alzheimer's Society, supporting early career dementia researchers across the world.
Hello, my name is Dr. Yvonne Couch and I am an Alzheimer's Research UK Fellow at the University of Oxford, and I'm excited to be taking a turn at hosting the Dementia Researcher podcast. Thank you all for tuning in for this week's two-part special discussing the 2021 Alzheimer's Research UK conference, which took place virtually this year. We recorded part one earlier this week, where I was pleased to be a panelist discussing the ECR day. And today I'm back, joined by 4 new guests to discuss the science, the panel talks, and the presentations from the week. This was my first specialist conference. I'm used to going to big ones that are just on neuroscience or glia, so it was a totally different experience, and I found the whole thing absolutely delightful. The balance of clinical research and fundamental science was awesome, and it was good to hear talks discussing everything from careers to the problems with clinical trials to drug discovery and design. So I'm delighted to be joined by 4 wonderful panelists today. We have Beth Eyre from the University of Sheffield, who you may know from her blogs on the Dementia Researcher website. We also welcome Dr. Lucy Russell and Dr. Aitana Sogorb Esteve, who are both research fellows at University College London, working in the Dementia Research Centre. And finally, Michelle Naessens, who is currently a research assistant at the University of Cambridge at the Centre for Frontotemporal Dementia. Hello, everyone, and thank you for joining.
Hi, everyone.
Hello.
Hi, everyone.
So before we start, I thought it'd be worth mentioning that all of us are new to the show, so be patient with us. I know we have all listened, but it's great to have a chance to take over and share what we've seen this week. First, let's do some brief introductions and talk about what we were at the conference for. Aitana, I know you had a poster at the conference, so do you want to start?
Oh, yes. Thank you for having me in this podcast. I'm a postdoctoral researcher at UCL, and I have a background in neuroscience and biology. And now I'm focused on fluid biomarkers, especially in genetic frontotemporal dementia. And while I had the poster for this conference that was focused on one of the aspects we look at the fluid biomarker team in my lab, which is the inflammation in frontotemporal dementia. And while my poster focused on a chemokine analysis in CSF, cerebrospinal fluid and plasma samples from some participants with primary progressive aphasias.
Excellent. So you must have really enjoyed the big biomarker session that we had the other day.
Yes, was great.
Yeah, it's good to see. It's good to see that there's stuff that you're interested in when you turn up at a conference and everything is unrelated. It's always a bit off-putting. Beth, you've mentioned you were short on time to get some data to present, but do you want to tell us a little bit about your work and what brought you here?
Yeah, for sure. So I was a bit of a lurker at the conference, which sounds a bit strange, but I kind of wanted to go to get a feel for like what's happening in the actual field at the moment, because my background's actually psychology, and I've only just started to move into the neuroscience. That's kind of what my PhD is doing. It's moving me over. So I'm currently investigating a mechanism called neurovascular coupling. I'm looking at this mechanism in the brain in Alzheimer's disease. And to do that, we use preclinical models. So again, this is all completely new to me, but I've really enjoyed the research so far. Obviously with the pandemic, using preclinical models, it took us a bit of time to like age the models, and then obviously the pandemic affected getting the data. So when the abstract submissions were supposed to be due, I wasn't like finished with my data, but I've started to collect a lot more. So I'm hoping that, well, by next year, I'll definitely be able to have a poster there.
A lurker is a great description. I was fully a lurker at this conference. And I've got a friend, Chris Martin, who's a neurovascular coupling person at Sheffield.
He's in the lab, yeah.
I was going to say, I'm assuming you know each other.
Yes.
He's a very lovely man. He taught me a lot of stuff during my PhD. So, Lucy, you had a poster in the frontotemporal dementia section. Can you give us a brief roundup of that?
Yes, so thank you very much for having me on the podcast today. So my work is focusing on genetic forms of frontotemporal dementia, along with Aitana. And in this group of people, individuals have problems with their behaviour and their language instead of memory, like in Alzheimer's disease. But because it's a particularly rare disorder, especially when you start to break it down into the different genetic causes, so that's expansions in C9orf72 genes and mutations in progranulin MAPT, we really need to tackle this on a global scale. And part of my role as the postdoc at UCL is working as the Frontotemporal Dementia Prevention Initiative Coordinator. And this is really linking big research groups from across the world together with pharmaceutical companies and consortium groups to promote clinical trials and really try and help design the best type of clinical trials that we can do for the cohort. So the poster was basically talking a little bit about that and some of the work that we've been doing on disease progression modelling in FTD, and comparing two of the main cohorts in FTD. So that's the Genetic Frontotemporal Dementia Initiative in Europe and Canada, run by Jonathan Rohrer at UCL, and also All FTD in the US. So it's looking really exciting data. The two cohorts look very similar, and we can see that individuals with progranulin mutations are particularly progressing much quicker than the other genetic groups. And so it's a really interesting, cool data coming out of it.
Excellent. It's really nice to see like these huge collaborative networks going on. And I think that some of the panel discussions during the conference really emphasized the importance of actually getting patients from all over the place, because, you know, if you have a very niche, I always feel that the niche patient cohorts we get in Oxford are people who are already kind of keen to be involved in research, and therefore, you know, their brains are not necessarily the same as the regular Joe out in the street who doesn't care. And I think getting a whole bunch of people from all sorts of places around the world is really important.
Yeah, I agree.
Good. Michelle, so you told me prior to this that you just slightly ran out of time to make your own poster, but you did say you were a co-author on a poster with Alistair Perry. Do you want to tell us a little bit about that and about your work?
Well, I'm a research assistant at the University of Cambridge. I work in the Cambridge Centre for Frontotemporal Dementia and Related Disorders. The lab I work in is quite diverse and quite big. So I've been involved on the GABA study and anti-tiagabine study, working on frontotemporal dementia and progressive supranuclear palsy to see if we administer drugs like memantine or tiagabine, if their brain activation, looking at EEG and MEG, become more similar to controls or not. So I thought that was really fascinating. So I work mostly on the EEG studies and MEG studies on frontotemporal dementia and progressive supranuclear palsy. But I'm also involved in our PET studies, where we look at inflammation markers, but also synaptic density in cortical basal syndrome and progressive supranuclear palsy. So it's been really nice getting to work with all these different patient cohorts and across all these different imaging modalities. So I'm looking forward to learning more, because my post will continue for another year. I actually finished my master's at UCL, and I did my dissertation in the same lab as Lucy. So it's actually really nice to come across a familiar face.
It's all nice and very inbred, but you'll find that as you progress in science, especially if you find an area that you love, an area that you're really interested in, you end up meeting the same people over and over again, especially when you meet old professors who've been around forever and they know everyone. And so suddenly everyone you know knows everyone else. It's very embarrassing. Okay, so I thought it might be good to do some day-by-day highlights to make sure we cover everything. As I mentioned up top, we covered the ECR day in part 1 of this series, so scooch back and listen to that if you'd like highlights from there. So we'll start here with day 1 of the main conference, where we talked a lot about novel drug design and inflammation. Beth and Lucy, do you guys have some highlights from day 1 you'd like to talk about?
I think it was a great start to the conference, actually, and the welcome introductions were really kind of thought-provoking and inspiring. And I think the ARUK video really highlighted some of the challenges that we've all faced over the last year with regards to the COVID pandemic and how it's been particularly difficult for those suffering from dementia. But it's also been really hard for us researchers too, and access to the labs. And even participant visits. And as you've heard from some of the others, collecting this data has been really difficult. But I think the video as well did a really great job of highlighting some of the achievements that we've made so far this year. And it's moments like this that we can actually all take some time to reflect on what we have actually been able to do over the last year and to keep us motivated with our work moving forward. And for me, it was particularly motivating to hear Shabnam talk about her own story and how her family coped with dementia. And I think moving on to some of the other talks for the day, Mike O'Neill gave a great overview of the current status of clinical trials in AD. And it's great for me to learn how clinical trials are being run in Alzheimer's disease to help us move into the FTD field. As I mentioned earlier, that's what I'm currently working on at the moment. So it's really helpful to see what things are working and what things perhaps aren't doing so well. I thought it was particularly exciting that the aducanumab trial is actually under review by the FDA and the European Medical Agency, despite the trial being stopped for futility back in 2019. And some of the other antibody-style drugs, such as donanemab by Eli Lilly, also showing some promise. And I thought it was particularly interesting looking at these two different trials here because of the difference in the eligibility criteria. The second trial by Eli Lilly really moved away from the clinical dementia rating scale as the primary outcome measure to a more kind of integrated and specific scale that might actually be better for measuring changes in kind of clinical presentation for the primary outcome measure. So I think that's really important to take away for all other studies and really thinking about which assessment tools we should be using in these clinical trials, and also thinking about the actual design of the trial as well and the inclusion criteria for the participants. And finally, I suppose with relation to my work in FTD, Mike also talked about some of the tau pathology trials in AD and other neurodegenerative disorders, such as progressive supranuclear palsy and FTD. And he was talking about a number of ways which we might be able to prevent the accumulation of tau. And this could be through the inhibition of tau expression or even by targeting post-translational modifications of tau, just to name a few. And he discussed the use of small molecule therapies and antibodies, as well as briefly touching on some antisense oligonucleotide and gene therapies, again, which are particularly pertinent to the world of FTD. And I think it's really exciting that we've got all these different trials going on. And whilst we do have some failures along the way, I think we're learning from these all the time, and we really can take from these and move forward, and hopefully we will have something useful in the next year or two. So yeah, I think it might go for a really great overview of where the field is at the moment, and it was a really great way to start the conference.
Yeah, and I thought it was a really nice. There was some, like you say, some disappointing news with the Huntington trial, but there was still a lot of positive messages in there that suggested that we can definitely go forward and make some progress in this area. Beth, what did you enjoy about day one?
So just to add to what you've just said, I really liked hearing about the clinical trials because it's not something I focus on too much because I am preclinical work, but I really appreciated when I think it was Mike, I'm not too sure, said how important the data collection at the preclinical stage is, because that's the sort of data that allows us to progress to these actual clinical trials in humans. So I really appreciated that. And that's something I'll definitely take forward with my own research. So the second part of day one was all about the prizes. The first two talks were from Dr. Sophie Bradley and Assistant Professor Renzo Mancuso, who they were both awarded the David Haig Early Career Investigator of the Year Award. And this is like, I think it's usually just given to one person, but I don't think they could choose this year. And it's worth $25,000 in research expenses, and they get a personal prize of $1,500. And I think they said that they shared the prize this year. And it's presented to the most outstanding early career researcher in the field of biomedical dementia research with less than 10 years experience post-PhD. So congratulations to those two. So Sophie gave us a bit of an insight about her career so far and some of the work she's currently doing. Her and her team are investigating the G protein coupled receptor superfamily. And their aim is to try and find potential new therapies for Alzheimer's disease through this G protein coupled receptor superfamily. The recent work that she spoke about is focused on determining how they can achieve these disease-modifying effects. And the results look really promising. I think they're using prion mice, and they used a M1 positive allosteric modulator. And the results suggest that by targeting this receptor, it can actually protect against the damaging changes seen in these prion disease mice. So I think that's really exciting for the field. It's not my background, but the way that she did the talk allowed it to be quite accessible to me, which was really good. And then the second talk was by Renzo, and this focused on his research, and that's using Zeno transplantation models to study human microglia biology in Alzheimer's disease. And again, his talk focused on some of the work him and his lab have been doing, and they basically developed a new method. This is the Zeno transplantation. It's a new method to allow them to study microglia and the genotype-phenotype interactions in Alzheimer's disease and these other brain disorders. Again, not my research area, but I did find what they were doing just fascinating. Some of the stuff that people can do now is genuinely, wow. I think science is just moving on and moving on. Things that we're going to be able to do in the future, it's really exciting. And then the final talk was from an early career researcher, Emma Jones, who was awarded the Jean Corson Prize for the best scientific paper in neurodegeneration research, which was published by either a PhD or an MD PhD student. And her talk was basically just about the paper. So the title of the paper was Identification of a Novel Risk Loci and Causal Insights for Sporadic Creutzfeldt-Jakob Disease, a Genome-Wide Association Study. So herself and the group she's in, they did a genome-wide association study and basically found 41 genome-wide significant single nucleotide polymorphisms, also known as SNPs, which I learned at the conference. And they were able to independently replicate these findings at 3 loci associated with sporadic Creutzfeldt-Jakob disease. So basically they were saying in the paper that this is like the first evidence been able to statistically see these robust genetic associations in this human prion disease, which implicate intracellular trafficking and some other mechanisms. So I think that was really exciting. Again, not my field, but the way Emma was able to do her talk did allow it to be really, really accessible for something that is so complex.
Yeah, it's definitely a skill. And I think being able to present something that is, like you say, potentially inaccessible is difficult. Or at the other end of the spectrum, I went to a presentation on a topic at some point a couple of years ago, which I fully expected to be really boring, but the guy made it so interesting, and you end up suddenly getting sucked in and going, oh, maybe I could do this. And then you've got 8 more research ideas, which you really don't have time for. Good. So that was day 1. Day 1 was brilliant. Let's move on to day 2. So day 2 is much more clinical. So I'm going to start with a basic sort of discussion type question for specifically Lucy and Aitana. So although Michelle, you feel free to jump in on this as well, because I know you work with people. So you guys work at a large dementia research center and you work closely with clinicians. How do you find, as sort of, I do not like the term basic researcher. I'm very against that. But as sort of fundamental researchers, how do you find that working relationship with clinicians?
I think for me, it's, I joined less than a year ago, this group on the DRC, and it's been a great experience because you have a very different point of view. And I'm indeed learning a lot about, in the case of frontotemporal dementia, about the cognitive, the clinical, and also the imaging. And I think that it's great because you create a kind of two-way feedback from the basic part to the more clinical part. And I think that's a very good thing to work with, because you take into account very different point of views and you can integrate it. And I think then you have a better picture of what you're looking at, basically.
Yeah, I agree. And I think it really makes you question every idea that you have, and how does it impact the patient? How does it help on an everyday basis? You know, I think we'll come on to later, but there was the sensory session looking at problems with vision and hearing. And those are the things that the patients have problem with on a daily basis. And those are the things that can make a huge difference. And I think sometimes it's quite difficult to see that when we're so focused on our own particular projects and we get our heads down in our books and our papers. And, you know, sometimes working with the clinicians, it gives you that ability to kind of take a step back and see the bigger picture and question why what you're doing is actually going to help. And, you know, you get to see patients, you get to work with them and hear their stories and go to support groups and do all those things that perhaps you wouldn't get to do if you weren't working with them.
Yeah, I think that the support group thing is something very new for me and very useful, because I've always been working in the lab and I only receive a tube with something and then I work with that, but I never see from where it comes and what is happening, what is the real life story of that. And it's really good.
Yeah, definitely. Michelle, do you want to add some stuff?
Yeah. Sorry, I completely agree with that as well, because I get to go to memory clinics as well. So you get to see how it actually impacts the patients, how it impacts their loved ones and their carers, because they sit in during these meetings and you just see their face go really sad when they underperform on cognitive tasks like these are. And it just shows you how relevant it is to do research. But we also, most of our clinical trials focus on clinical trials and treating the disease. But in this next session, Craig Ritchie also mentioned that we should look at symptomatic treatment and actually do trials on those, because treatment for the disease is amazing, but it's so far ahead in the future still that we should also look at how we can actually maintain the quality of life of the patients that have the disease right now.
Mm-hmm.
Yeah, that's definitely really important. It is something that's coming in a lot of fields. So my background is stroke, and the issue we have in the stroke field is people have tended towards, almost in the same way that they've done in the dementia field, you look at the brain and is your intervention fixing something in the brain? So in the case of dementia, or, you know, is it reducing amyloid? Is it reducing tau? If it is, then it must be great. And it's the same with stroke. Is it making the infarct volume smaller? And actually the field's moving away from that now to saying, actually, are the functional outcomes there? So, you know, in terms of preclinical research, if you've given an animal a stroke, can it walk better after your intervention? You know, can it use its limbs better after your intervention? And that kind of stuff, like you say, in dementia is really important, because it is all about getting the person's quality of life back. But that leads us really well onto the big panel discussion they had on day 2 about clinical trials. And Michelle, you just briefly touched on it there. Do you want to give us a bit more information on the sorts of things that they discussed?
Yeah, that clinical trial session was absolutely fascinating to me. And I think what I really liked about it was the need for representation in research. I think Lucy knows that well, that the patients we recruit from our studies get recruited from specialized clinics. So our movement disorder clinic or a memory clinic. And those people presenting to these clinics, they all are already a biased group, let alone those that then go on to go into research or even clinical trials. So when you look at our cohort, FTD cohort, these people tend to be highly educated from a higher socioeconomic status, and they also tend to be white. But that is not representative for the whole UK population or the patient groups in general. So I think that is really important to look at. And when you look at our research, I conduct research in Cambridge, and of course that can never be representative of the whole UK, because Cambridge is a special place in and of itself. So I think that is really important to take into account where you're doing research. Both Cambridge and Oxford, they are very special places, even though we're rivals, but we will have biased patient groups in our research. So that is very important to take into account when you're doing clinical trials. So I think Craig Ritchie, again, he mentions an opt-out system for research, same as you do with organ donation, which would be really good. But that also depends on those people already presenting to these specialized clinics. So I think we should start before. And I think the Joint Dementia Researcher Joint Dementia Research is a really great platform for that. But I think we should also work in the community and go to these people, because once again, those people that go on these sites, they are a bias group. So I think there's just so many more things we should take into account.
Yeah, very much so. It's something that we have a problem with preclinically. I know in that we almost do the same thing. You know, we do all of our experiments on young, healthy, male mice, and that's just not what any of this is about. These are all aged populations. They're mixed. They have comorbidities, and so we have to link it up all the way throughout the field. So we have to get our preclinical research populations appropriately mixed, and then we have to get our clinical populations appropriately mixed if we're ever going to think about translating any of this stuff.
I completely agree with, sorry to interject, but I completely get that, especially from being at the preclinical level. A lot of the labs, like you said, just use male mice. I've said I wanted to use male and female mice because dementia also affects women. And it's the same with the clinical trials. A lot of the data, it's from males. And how can we use the data from the clinical trials if there's not the right gender split? And again, same with economic status and ethnicity and things like that. And it is really important to get that representativeness. And you mentioned about comorbidities. Most people who have like the symptoms that are dementia, it's not just one thing that's causing them. It's one or two things, isn't it? So in our lab, we also research comorbidities. So we have like Alzheimer's and we induce like atherosclerosis as well to look at the links between Alzheimer's and heart disease, that sort of thing.
Yeah, and having these kind of platforms where you can access different patient cohorts, like Lucy was talking about at the beginning, I think are going to be really important going forward. So in terms of big networks, we also on day 2 briefly discussed the Eden platform, which for those of you who don't know is the Early Detection of Neurodegeneration Initiative. They have a website, go check it out if you're interested at edeninitiative.org. Beth, I'm wondering whether you could give us a brief roundup of what the Eden Initiative is and why it's important.
Yeah, so the EDON, or the Early Detection of Neurodegenerative Diseases, is a really exciting project, actually. And their overall aim is to detect these neurodegenerative diseases way before we see the symptoms. With the things that cause dementia, with the diseases that cause dementia, you see the symptoms when the disease is really quite progressed. And obviously the point of this is to interject early, which could potentially stop people having the symptoms, which really do affect an individual's life. So if this works, it could be really life-changing. And the whole point is they've got together a big group. It's a big collaborative project globally. They have data scientists and digital technology scientists, clinicians, and they're all working together to create a tool, a digital tool that includes like wearable technology and smartphones that potentially could help with the detection of these neurodegenerative diseases. And the point of this digital tool is that they want to collect like vast amounts of data and then try and link these with clinical tests such as brain scans in order to see if there's like digital patterns, a bit like a digital like a fingerprint, to see if they can like identify fingerprints in different neurodegenerative diseases. And again, like I mentioned, if this does work, it's really exciting because it could really change people's lives.
Again, it's all about catching stuff early and having that huge network of available people. And with so many neurodegenerative diseases, Alzheimer's, Parkinson's, whatever, you don't catch it until you've already got it. And so these kinds of studies are so important. One of my favorite ones is not Alzheimer's related. It's the woman who can smell Parkinson's. Still officially my favorite paper ever. So on the third day, which we'll move on to now, we saw some heavy hitting and timely topics coming up, starting with some work on the influence of viruses on dementia, particularly important this year. And we followed up with some work on biomarker discovery. I'm going to leap in because for me, the work of Colm Cunningham is always a pleasure to see. He's interested in peripheral inflammation and how that affects the brain and some communication pathways between the brain and the periphery. And he uses some very elegant models, and he just always presents his work very beautifully. So I love Colm. So that's my highlight from day 3. But Aitana, is there anything you really enjoyed about the viral sessions?
Yeah, well, I think Colm talk was particularly interesting. And I think, inflammation is a topic that increasingly has more importance in dementia. There are increasing evidences that microglia plays a crucial role. And from Colm's talk is that this systemic inflammation can also affect the brain, of course, because it's something systemic. But I particularly found the first talk from Clayton Wiley very informative. There was a couple of things that I didn't know about this relation between dementia and viral infection. A couple of things that were commented in that talk is that even there is not a real relation between infections and dementia from the literature. It seems that just the fact of the aging, to have an old immune system, that is something that can be a risk for developing something more after viral infection. And also 2 more things that shocked me is that there was a fact that aerosol transmitted virus they can affect more the nervous system, because if they are transmitted by the air, the immune system cannot tackle them properly or better than other infections. And also another thing that was commenting, this was just an hypothesis from that was discussed in the questions, and it was the theory that maybe the microglia activates this synaptic protein to avoid the transmission of the virus from cell to cell. And I think that's something I have never thought about it. And I think it's a very interesting topic, a very interesting theory. And then the session about the COVID-19 was very interesting for the times that we have now, and just the fact that Marcus found viral particles in the vagal nerve. It was very shocking for me. And then, of course, there is this increasing evidence I mentioned before, and that was something that Jarvis explained in his talk about the gene association with Alzheimer's disease and this COVID-19 severe people. So yeah, I think it was all very interesting and on topic now.
Yeah, definitely. There was one bit of that session where I had to look. So genetics is not my field at all. There was something about quantitative trait. I'm going to go with Beth's version, which was loci, but it could be loci. I'm never sure how to say that word. But that I had to look up, and I was like, ooh, you can get different amounts of mRNA expression at different people can have different amounts. This is so cool. And I feel that that's very like first year undergrad genetics, which I was either asleep for or I'm so old that it was discovered after I did my undergrad. So yeah, that kind of stuff where they were looking at the severity of SARS and the genes that are expressed and how that might relate to aging. I thought it was fascinating. And the fact that you can have viruses, potentially have viruses repeatedly throughout your life, and this might affect how you age, which makes me a bit paranoid about taking public transport, but I don't think there's anything we can do about that. So, Michelle, you use MRI as a way of looking at dementia, but do you think combining it with other biomarker studies would be useful? And was there anything from the biomarker aspect of the day that you thought was particularly inspiring?
Yeah, I think you can't do research without pooling your data from different types of biomarkers. Because I love MRI. It's the imaging method I have most experience with. But there's only so much you can tell with it. Like Nick Fox said during his presentation, MRI is great for diagnoses. If there's atrophy of the hippocampus, you can quite confidently say that this person has Alzheimer's, or if there's amyloid deposition in certain parts of the brain in a PET scan, you can also say that person has Alzheimer's. But it's a quite static marker, so you can't discern small changes over time. So it will never be able to tell you if your treatment is successful, if there is a trial happening. So I think what is very important is to look at markers that are dynamic, that can detect these little things over change. So blood biomarkers or CSF biomarkers would be great because they're quite easily obtained. You can take them multiple times without really having an effect on the patient. As if you do a PET scan, that's radioactive. So there's only so many scans you can do. And they're quite costly as well. So if you take blood biomarkers, you can analyze a lot of them in one go. And it's quite cheap. What I thought was really interesting was this salivary biomarkers. Because I knew that a lot of labs took saliva from their patients, but I didn't know how they analyzed them or what the findings were, how far the field had progressed in that regard. So I think that would be amazing as a dynamic marker of change. And it will be really great for a patient as well, because they can just take their own saliva and then just send it off in a post box even. They don't have to come into clinic. And quite similar to an at-home COVID test, really. Just put it in a post box and just go on with the life. Additionally, I thought the digital biomarkers were really interesting, so tying it in with the Eyre network. I thought the in-ear EEG analysis was really amazing. I didn't even think that was possible, so it really blew my mind.
I didn't realise there was something like that existed. I think that compared to the sleep headwear that they were suggesting, which is a brilliant idea and concept, but in reality, I'm not sure it would work that well, particularly when you think of behavioural wearing FTD patients. Like, there's no way they're going to keep that on their head for a night. Whereas those little in-ear EEGs were incredible.
Yeah, it just really blew my mind that was even possible. How do you even come up with that idea? So yeah, just there's so many things happening nowadays, and technologies advance so quickly. And there's so many people out there in the field that are just so incredibly smart, developing these technologies, developing these methods. It just gives me so much hope for the future. So I think if we can pull this all together, like these different technologies, different methods, different biomarkers, and then working across different studies, different centers, different countries, I think the sky really is the limit.
And I think, like you say, so like applying some of those techniques that are just so snazzy, like that in-ear EEG business, really will help with patient recruitment. So if you can give if people don't have to come, people don't like hospitals. If they don't have to come into a hospital, or they don't have to come into a funky study center and sit in a clinical waiting room, when they've got dementia, and that's going to make them uncomfortable and they're going to get anxious, and, that might affect your design. If you can get them in their natural environment, then, it's so much easier. Aitana, you work on biomarkers. Did you enjoy that session as well?
Yeah. It was great. Yeah, and also it has been lately a lot of updates on the fluid biomarker field as well. So I think Henrik's talk was great, mainly on this blood biomarker, blood phosphatau, because the problem is that blood is not being easy fluid to find biomarkers, because for dementia you need to find things that reflect the state of the brain. And sometimes the concentration of those biomarkers in blood is very low, that you cannot detect it with any technique. So, but the research is now focused on that, and I think there are very good improvements and developments on that.
Yeah, and like Michelle says, I think if we just all pull together and really focus our efforts, I think spectacular stuff could happen. So finally, we'll move on to day 4, where we had some important talks on protein misfolding, but I'm going to steal the stage a bit here and talk about Dr. Rachel Waller's talk from the first session, which was on the development of dementia post-stroke, which is my field of fun. So she was using the COGFAST cohort, which is a clinical cohort. It's run by Raj Kalaria in Newcastle. And what she was doing is she's taking postmortem tissue and she was using laser capture microdissection, which I think is a beautiful and slightly underused technique. And she was taking out endothelial cells and astrocytes and neurons, and then she was doing microarrays on each cell type. And what she found was that she got this fundamental breakdown of the neurovascular unit, but this was at a site that was distant from the stroke. And for me, that's one of the important things about vascular dementia after stroke, is that your stroke often happens in the back/middle of your brain, and you can have all of these vascular changes that happen distant from this site. And there doesn't seem to be any link between the size of the stroke or where the stroke is in terms of who is developing vascular dementia and who isn't. So something else has clearly got to be going on. And I thought that Rachel really started to pin down some of what was going on there. So I thought her research was really fascinating. But then we had the neurosensory section. So Lucy, can I come to you and ask whether there was anything from that you found particularly fascinating?
Yeah, I think particularly the first two talks of the session, the one by Maya and then the other one by Jason Warren, were particularly interesting, looking at these things that kind of affecting the patient's everyday life. So the first one was about using the retina to predict status of Alzheimer's disease. And she was talking about how we can actually find changes in the retina very early on in the disease. And actually we could be looking at things like tau and amyloid within the retina using very relatively cheap imaging tools. And it could be an alternative way to pick up these things in everyday life. You know, if you went to Specsavers for your eye test or something, you know, it could end up being something that's actually a practical tool and a screen in a clinical setting that could be useful very early on. And this was kind of iterated in Jason's talk afterwards around hearing and developing a hearing screen that could be used in clinics to pick up these early changes. And he was talking about some of these difficulties that often individuals with Alzheimer's disease say that they have in terms of really kind of getting lost in social situations, and this background noise really kind of taking over and making it very difficult to focus on the conversation that's at hand. And they've done a variety of different tasks in hearing, from kind of peripheral hearing problems and musical hearing, and he kept referring to the cocktail party effect that, like I spoke about before, and I think these sorts of things can be really useful down the line. And yeah, I think they were fascinating talks and something that definitely need exploring further to see what utility they can have in everyday life to pick up these diagnoses earlier.
Yeah, and it harks back to that, you know, being able to recruit more people at easily accessible places. You know, everybody has eye tests and loads of old people have hearing tests. If you can catch them there, that is nothing but a good thing. So I know that we're short on time, but it was a digital conference. It was a virtual conference. And I personally thought the platform was really interesting, but I wanted to ask you guys what you thought about it. So Beth, should we start with you?
I quite liked it, actually. This is my first proper virtual conference. I've not really had any experience with the others. And I hadn't really been to any real conferences before. So I have nothing really to compare things to. So I guess that's quite good. But I thought it was really accessible. I liked that you could chat to other delegates. I had a couple of conversations with people, and I really liked the on-demand section afterwards. So if you did miss a talk, because I had to miss one or two because of things I couldn't get out of, it's nice to know that it's still there and I can go and catch up on it.
Yeah, definitely. And Michelle, you're going to be relatively new at this whole conference game because you're sort of much earlier in your career than all us old people. How did you find the platform?
Yeah, I really liked it. The setup was quite minimalistic, so it was quite easy to find your way, find when things were being presented, where to find the posters. So I went to AAIC in summer, and I thought that platform was way more complicated and more complicated to move across because I couldn't really find things. So I just chose to watch things on demand. And I like this a lot better. What I would have preferred to be there was closed caption options to the videos. Maybe they are there, but I didn't see them. I think that would help accessibility.
Yeah, I agree with you on that front. I do, although I do occasionally find them distracting when they start to miss, especially when people have accents, when they start to mistranslate what they're saying, and then you just get distracted looking at the funny sentences that are clearly not what the person just said. But if it's done well, it's really helpful. So Lucy and Aitana, have you done many virtual conferences over the last year? How did you find this compared to the ones that you've already done?
Yeah, we've been to a few, haven't we? I thought it was very good on a whole. I thought that, in general, the platform ran well. I did have a few issues with it freezing. I don't know if other people did on the live video. Yes,
definitely, repeatedly.
Frustrating, but it was a quick fix. A refresh of the screen, and it was back. Perhaps in my old age, I would have perhaps liked a PDF of the agenda. I liked the interactive style of the agenda and the way you could bookmark the sessions that you liked. But when I first got into the conference at the start, I just wanted to have a quick glimpse through what was on and the clicking between the pages. But I mean, it was a minor thing. It worked perfectly well. But yeah, that would have been the only thing I would have liked.
Yeah.
Aitana, how did you find it?
I agree with Lucy. I mean, it was great. And I really liked the way that you go to the day and you can see all day and you click and you linked directly to the talk. But yeah, I missed maybe a PDF agenda as well. And also just one thing that have driven me a bit crazy is the poster notifications. So if you got any comment, you have to tag your poster because, well, maybe it has something only happened to me, but I couldn't get any notifications that people was asking me things in the poster and I had to tag every time. But for the rest, I think it was really good. Yeah.
Yeah, it was a nice platform. Go, Lucy, you've got something to say.
Yeah, another thing I really appreciated, and thank you ARUK for doing it, is the breaks. I have been to a number of conferences that have slammed you from 5 hours at a time without a break. So I really did appreciate those. So thank you.
Yes, we've got lots of nodding from all the other panel members, and I went to one earlier in the summer where they did flash presentations. And don't get me wrong, I love a flash presentation, but these were all 5-minute talks. They'd done it poster style, so you could barely see anything. And they did about 15 of them in one go. And I got to about 7 and I was like, I don't know what's going on. My brain hurts. So yeah, repeated breaks. And the fact that they were half days so that you could spend the other half of the day doing, science or childcare or whatever you're, looking after your cats, in the case of Selena, whose cat repeatedly pops up on Twitter, which I love. But yeah, it was a really nice layout. As a new person, I really enjoyed the disturbing, dating app style matching, who's because I found someone who was doing stroke and vascular dementia and inflammation. I was like, oh, let's be friends. It was really childish. Michelle, did you want to leap in?
Yeah, there's another thing I really liked was that the on-demand session was available next day. Some conferences, they wait until the whole conference is done, but especially since it was only a half day, I could just choose to watch things back the next day, which was really nice.
Yeah, there was plenty of space, which was lovely. Well, thank you everyone for sharing your thoughts. So if you attended the conference and missed those talks, we hope you can seek them out and watch them on catch up. If you didn't book and can't access those presentations, I hope our summaries today were interesting. And if you check out Twitter with the hashtag ARUKConf21, you will find plenty of chat about the week. So before we finish, I'm gonna let all of our panelists give a plug to anything they might want to. So let's start with Beth. I know you've got podcasts and Instagram going, so do you want to plug them?
Yes, so if you want to hear about my experiences as a PhD student and some of the research that I do, you can follow me on Twitter and on Instagram, which is where I do most of my science communication. And that is at Beth's Brain Bites. And I also have a podcast. The podcast is called Honest Academia. You can find it on Apple or on Spotify or on other places you get your podcasts. And myself and my friend Mal, we talk about the experiences of navigating academia as first generation PhD students.
Excellent. Thank you very much. So, Lucy, you've mentioned to me that you've got a student working on something similar to the Eden project. So if you give us a bit of a rundown of that, and then Aitana can run through your social media and the FTD Talks website stuff.
Yeah, so one of the students that I'm helping to supervise, Rhiannon Convery, she's doing a program very similar to Eden, but in frontotemporal dementia. So it is also called the Early Detection of Frontotemporal Dementia, or known as EDOF. And you can find her poster if you're at ARUK in the frontotemporal dementia session, and it was number 4.8. As part of this project, she has an iPad app called Ignite, and that's IGNITE. And she's currently validating this in the general population. And so far, she's done really well and managed to collect data on people aged 20 to 80, and is hoping to get 100 men and 100 women from each decade. At the minute, she's actually recruited just over 1,500 people, which is an amazing achievement. But she hasn't quite got enough individuals under 50 to have taken part in it. The app's made up of a few fun, short activities, but like brain training games, and it really doesn't take very long to complete. So if anybody would like to help, please download the Ignite app from the Apple App Store, or you can check out the website www.igniteftd.com, and you also can find her on Twitter at EDOF.
Perfect, thank you. Aitana, do you want to give us a rundown of your stuff?
Yeah, of course. Well, both Lucy and me are part of the FTD Talk team at UCL. And in our aims is the public engagement and raise awareness about genetic FTD and FTD. So we have a website, it's ftdtalk.org. And there we have some useful information about FTD, about our research. We also have a blog in which we upload our latest research and some interesting posts. And then we also have a Twitter account, an Instagram page, and a Facebook page. All of them FTD Talk. So if you follow us, we post some information about FTD, genetic FTD, and our research.
Brilliant. Thank you ever so much. So, Michelle, you're still relatively new. Do you have anything that you want to plug?
So nothing really majorly research related like the rest of you. But during lockdown, I've really embraced my inner grandma and I've started embroidering. But since I don't want to end up with loads of embroidery things, I decided to start fundraising for Alzheimer Research UK and Into Science UK. So I have an Instagram page, Michelle Embroiders. And then you can ask for commissions or just buy pieces I have, and half of the profits will go to Alzheimer Research UK or Into Science UK.
That is absolutely fabulous. Well done on setting that up. And as someone who is funded by Alzheimer's Research UK, I would like to say thank you very much. It is beautiful that you're doing that. So it's time to end today's podcast recording. I would like to thank our panelists, Beth Eyre, Dr. Lucy Russell, Dr. Aitana Sogorb Esteve, Michelle Naessens, and if everyone can say a big goodbye.
Bye.
Bye.
So we have profiles on all of today's panelists on the Dementia Research website, including details of their Twitter accounts. Thank you all for listening. The Dementia Researcher podcast is available on Spotify, Apple Podcasts, Stitcher, or wherever you get your podcasts from. And please remember to like, comment, and subscribe. And keep sciencing, everyone.
Brought to you by dementiaresearcher.nihr.ac.uk in association with Alzheimer's Research UK and Alzheimer's Society, supporting early career dementia researchers across the world.